Hi everybody. Title of my talk today is Develop Novel Therapy to Prevent Vision Loss Among Older Populations. So overall goal is to develop novel therapy to prevent vision loss. I think everybody will agree with me that how important vision is. We rely on this special sense to engage with the world to see around us. It develops the relationships and reacts to the sight of unique building, flower, animals, trees, and your own people. Most people around the world agree that it is one of the senses that people fear to losing the most. Can you imagine gradually losing the ability to see? Unfortunately, there are 11 million Americans suffer from early form of this disease called age-related macular degeneration, and already 2 millions have lost their vision because they have the devastating late form of this disease. AMD is an incurable disease, most commonly seen in those over 50. So it is becoming increasingly common when people age. So 1 out of 5 Americans are over 50 and 1 out of 4 individuals over the age of 65 develop this disease. As the aging population increases with a better lifestyle, it will continue to rise, making a critical public health problem because it affects the older population. Normally, we are able to see as light sensitive cells from the eye take the light around us and then send the light in the form of electrical impulses to our brain, allowing us to perceive the images that surround us. The light sensitive cells are called photoreceptor cells and are supported by underneath epithelial cells that supplies oxygen and nutrients. So this is very important because those photoreceptor cells basically they are supported by those cells we call as epithelial cells, or sometimes it's a retinal epithelial cells. Vision loss in AMD patients is caused by degeneration of these light sensitive cells and underneath supporting cells within the eye. So when the supporting cells for the light sensitive cells die, then the eye no longer have support and nutrients to maintain the good sight. Without that support, the light sensitive cell starts to degenerate. Then the vision loss occurs. Vision loss in AMD patients is characterized either as a dry AMD or neovascular AMD. So the advanced form of dry AMD, also termed also geographic atrophy, is caused by death of those light sensitive cells and underneath epithelial cells or supporting cells. In a neovascular form of AMD, basically what happens, the blood vessels from back of the eye, they grows into the eye, invert into the eye, causing vascular leakage and scarring within the retina. In either of the forms, central vision is severely affected, leading to loss of independence and loss of your own function. Even though treatments are available for wet AMD, the neovascular form of AMD, there is no effective treatment for dry AMD, though research on supporting cell replacement therapy is some progress. So when we'll study about the mechanism of how these things are developing, so to focus on that, what we see that in like damage to the light sensitive cells and supporting cells has been linked to progressive vision loss in dry form of AMD. AMD is a multifactorial disease having metabolic, genetic, and environmental components. So generally we need oxygen to live. High concentration of it are actually corrosive and toxic. So we obtain energy by burning fuel with oxygen. That is by combining digested food with oxygen. From the air and we breathe. This is a controlled metabolic process that helps in the body metabolism. But unfortunately, it also generates dangerous byproducts. These includes free radicals, that is electronically unstable atoms or molecules capable of stripping electrons from other molecules when they were meet or when they contact or in touch with them to achieve the stability. So if free radicals are not taken care, it can chemically interact with the biomolecules such as DNA, protein, lipid, and different membranes, causing a serious damage to the cell. We have our own antioxidant defense system against these oxidative stress related changes in the form of physical barriers to contain free radicals at their sites of production of within cells, and also enzymes that neutralize dangerously reactive forms of oxygen, and substances in our diets like vitamin C, vitamin E that can quench free radicals by donating electrons to them and cutting off the chain reactions in their core so that they won't damage those biomolecules. So this repair mechanism, basically the way it works, they can take care by damage from the oxidative damage to the DNA or proteins or different membranes, if it doesn't taken care, then it leads to stress related or programmed cell death. So in response to AMD, animal models of AMD and some age-related eye disease clinical trial study strongly support a role of oxidative stress in the development of AMD. Because of the importance of oxidative stress as a contributing factor in AMD, treatments that reduce oxidative stress may be valuable as a therapeutic, though antioxidant therapy has shown more benefit in preventing progression to wet AMD than to geographic atrophy. So today I am going to talk about the animal model of dry AMD having visual dysfunction and how gene and drug-based therapy could prevent visual dysfunction in this model. So our lab has developed a mouse model of dry AMD in which we see elevated oxidative stress in the eye, causing death of light-sensitive cells and underneath supporting cells. We found markers of oxidative stress elevated by 2 months of age in this model. We used similar tests used in the clinics to measure retinal function and evaluate retinal structure. In the mice model of dry AMD, we found significant reduction in retinal function and also significant reduction in thickness of retinal outer nuclear layer after 6 months of age. Using this model, we study the mechanism of retinal degeneration and develop new therapy to prevent vision loss. We have published research demonstrating repurposed drug and antioxidant gene therapy to improve retinal function and structure in this model. First, I'll talk about gene therapy and later I'll talk about drug therapy. So gene therapy is an experimental technique that uses genes to treat or prevent disease. In the future, this technology or this technique may allow doctors to treat a disorder by inserting a gene of interest into patient cell instead of using a drugs or surgery. Gene therapy is designated to introduce genetic material into cells to compensate for abnormal genes or to make a beneficial protein. A gene that is inserted directly into the cell usually does not function. Instead, a carrier called a vector is genetically engineered to deliver the genes. Certain viruses are often used as the vector because they can deliver the new genes by infecting the cells. So these viruses are modified so that they don't cause any disease when used in people. For antioxidant gene therapy, we injected 1 microliter of an antioxidant gene therapy vector into the mice at 6 weeks of age, causing early treatment, and waited for 9 months to see its effects. The idea was to produce antioxidant enzymes in the eye tissue constantly using the animal's own biological system. So what do we see that antioxidant gene expression due to the gene therapy was measured, and we saw that there is a reduction in oxidative stress after 1 month after injection. In response to our treatment, our gene therapy treatment, what we see that we saw loss of visual function was restored and structural integrity was significantly delayed. Basically, it doesn't degenerate until 9 months. When we did the retinal histology, we found better retinal ultrastructure compared to untreated ones. We also tested another drug called 8-hydroxy-DPAT, which has been approved to treat anxiety disorders in other patients. This drug is demonstrated to prevent oxidative stress in the eye. So we systemically delivered daily 3 different doses, around 5 mg per kg, 10 mg per kg, and 30 mg per kg of this drug for 4 months. We see the higher dose of this drug was able to reduce oxidative stress compared to the other 2 doses. After 4 months of treatment with this drug, we found increased retinal function and preservation of retinal light-sensitive cells and underneath supporting cells. To conclude my talk, in summary, In summary, what we see that complementation of antioxidant gene therapy and daily treatment of 8-hydroxy-D-PAT, that's the drug, slowed down retinal degeneration in the mouse model of dry AMD. And our research supports potential antioxidant gene and drug therapy to prevent visual dysfunction that is commonly seen in older persons. Our research also raises some important questions to investigate mechanism of visual dysfunction for the future research. Thank you for listening my talk. Please, if you have any question, please contact me through my email, which is visual@ufl.edu. Thank you very much for listening.